Okay, welcome to Let's Get to the Marks. I'm going through GCSE biology. This is the 2020 paper one higher. There's 7 questions, total of 100 marks. So, yeah, 140 minutes roughly to do this paper. Let's get into it. Okay, question one. This question is about photosynthesis. Complete the word equation for photosynthesis. So what I suggest you do, grab a pencil, grab some paper, go get it now. Grab your calculator. You're going to need that in a GCSE science exam. Press pause, have a go, unpause, watch the walkthrough. You know the situation. Remember, the exams are coming. We've got 2 weeks. You got to get on it. Let's go. Okay, complete the word equation for photosynthesis for 2 marks. So when you're wondering about this, just ask yourself, what do plants take in? Photosynthesis happens in plants. What do they take in? And two of the things they take in are water, and we also have carbon dioxide. Now, these are the reactants for photosynthesis. Yes, you need light, but light in GCSE terms isn't made of particles that are actually like atoms, like these things. So light would appear above the arrow. Anywho, we don't need to put that here. So glucose. And there we go. They take water, they take carbon dioxide, they rearrange it, they make glucose, and they release oxygen. Describe how energy for photosynthesis reaction is gained by plants. So how is it that plants get the energy to overcome the activation energy and get the water and the carbon dioxide to collide successfully to react? And the idea is they absorb sunlight. Now, specifically, you might want to say UV light is what they actually need. And where is it in the plant that this is absorbed? In the chlorophyll. Oh my gosh, I can't spell. Serious issues with spelling. Chlorophyll. Now, they may allow you to put chloroplast. Specifically, it's the chlorophyll that absorbs the sunlight. It actually releases electrons in quite a complex reaction. So, chlorophyll absorbs sunlight, and that is where plants get the energy. So, there we go. First 4 marks on the board out the 100. All right, let's move on quickly, quickly, quickly. We've got lots of revision to do. Studies investigated the effect of temperature on the rate of photosynthesis. The students shone light from a lamp onto pondweed and measured the volume of oxygen produced per hour. Okie dokie. Table one shows the results. So what we got going on here in table one is we've got the temperature. So they've obviously changed the temperature. That must be our independent variable, 20, 25, 30, 35, 40, 45. They've done 3 repeats at each temperature, and they've calculated the rate of photosynthesis in centimeters of oxygen per hour. Obviously, they've put an X here. You can guess what they're going to ask us. They will ask us to calculate that mean. Let's go, let's go, let's go. Calculate the mean X, 2 marks. You couldn't get much easier than that, could you? So what do we want to do? When you calculate the mean, add up all the individual repeats, divide by the total number of repeats. So we want to do, we want to smash this in the calculator. 18.5. We want to add that to 19.3. We want to add that to 19.5. Then we want to bust out the divide line or press the divide in your calculator by 3. And what does that equal? That equals 19. Let's go to one decimal place, zero point 0.1 centimeter cubed. And that is per hour. So we can write that as in per hour. Lovely jubbly. Okay, and that's going to get you 2 marks for calculating X. Oh my good gosh. Students identified an anomalous result. Do you know what anomalous means? It means odd. I feel a bit anomalous sometimes, a bit odd. But so anomalous means odd. Which is the odd result in table one? Draw a ring around it to identify it. That gets you a mark. And then suggest one reason why we might have an anomalous result. Why is it the students might have got an anomalous result and the question is a suggest. So we'll have a look at that too. Let's go up and find this anomalous result. Um, let's go. Yes. Okay. Let's scan read. So 18.5. The way to spot an anomaly is compare the repeats. These are quite similar. These are all quite similar. These are all quite similar. 38.6, 39.8. Yeah, that's quite far out, but still pretty similar. Let's see if there's any worse ones than that. 44, 1.9, 14.2, 2.2. Oh, obviously these 1.9 and 2.2 are similar, but it's fairly obvious the 14.2, when you compare to the other repeats at that temp, is an anomalous result. So that's going to get you 1 mark for slamming that down. Now, what's the reason why they might have messed up and got this anomalous result? So what we can do is maybe we need to look at the method to find out where they've gone wrong. They've obviously made a mistake in the method. Let's see if they gave us any instructions on the method. Not a lot. Students investigate the temperature on rate of photosynthesis. The students shone light from a lamp onto pondweed and measured the volume. Right. What haven't they told us about the light from the lamp? They should have kept the light the same each time. Maybe someone moved the light too close here and they moved it really close and you got loads more oxygen given off at that temperature. So maybe they didn't keep the distance of the lamp the same. Um, Maybe someone didn't connect the gas syringe correctly. Maybe someone didn't use a water bath to keep the temperature the same and the temperature wasn't controlled. Yeah. Maybe they didn't put the same mass of pondweed in for that exact experiment. Maybe they used a much bigger amount of pondweed, which produced a lot more oxygen. So there's all sorts of suggestions. My main one from looking at the method that you would come to, I think, is that they didn't control the distance of the lamp. So that is what I'm going for. They did not control the distance of the lamp, and so the light intensity changed. Maybe they added too much hydrogen, sodium hydrogen carbonate that releases carbon dioxide. There's all sorts of mistakes they could have made, but they definitely didn't control all the variables. So that's going to get you a mark. How did the students deal with the anomalous result? Right. What we need to look at is, what did they do? I know what they should have done, but did they do it? Let's go check if they did it. You know how you should treat an anomaly? You should treat it very badly. What you got to do with an anomaly is cut it out and calculate the mean without the anomaly. And I believe they've done the right thing, because look, 1.9 plus 2.2, what's that? That's 3.2. That's like whatever the hell that is. 4 point, what is that? 3.2, 4.1, something like that. So it looks like they have, and then divided by two. Yeah, they've done that. So they've taken the anomaly out of the mean calculation, and they've done 1.9 plus 2.2 divided by 2, which is like 2.05, and then they've rounded to 2.1. So what they did with the result is, they did not use it in the mean calculation. And you can see that, right? Just to show you that they did like 2.2 plus 1.9. What does that equal, folks? That equals something like 4.1. They then divided that by 2 instead of dividing by 3, because they removed the anomaly. And then it got something like 2.05, and then they would have rounded that to 2.1. So that's what they did. You do not need to show any calculations. You just need to say that they didn't use it in the calculation of the mean, which is what you should do with anomalies. Give one factor the students should have kept constant. Right, there's a whole bunch. We just talked about how they possibly screwed up. What should they have kept constant in this investigation? If you're changing temperature, do you know the two other um, limiting factors of photosynthesis? So you've got temperature. Okay, so they should have controlled CO2 concentration. That's one of the things. They should have controlled light intensity. You can do that by using a ruler in the distance of the lamp. CO2 you control by using the same amount of hydrogen, sodium hydrogen carbonate or bicarbonate of soda each time you put the same mass in. They should have also controlled the mass of pondweed. Right, you want the same amount of pondweed, the number of leaves on the pondweed. Very hard to control the mass of chlorophyll in there, but yeah, you do your best. Any of those is going to get you the mark. I reckon CO2 or light intensity is the best. Don't say control temperature, because it's obvious, right? If you said control the temperature, give yourself a quick slap in the face. Because look, it's obvious that they were supposed to change the temperature, right? The temperature was the independent variable. Independent variable is always found in the first column in a table. Make sure you remember that. And it's obvious that is what they're changing. You're always changing one of the variables, and you should be controlling the others. Right, let's move on. Ooh, table one is repeated. Why did the rate of photosynthesis decrease from 35 to 45? So after 35 degrees, the mean rate of photosynthesis is going down. What's happening in there? Hopefully you guys know what's happening in all living things. Every chemical reaction in a living thing is controlled by enzymes and substrates. However, at a certain temperature, what happens? The active site denatures and the substrate will no longer fit into the active site like a lock and key, and therefore the rate of reaction decreases because enzymes are biological catalysts, guys. So what we want to put here, I'm going to put, I'm just going to put because, whoa, enzymes denatured. Yeah, if you do biology A level, you learn all about the enzymes that are in control of photosynthesis, such as rubisco. So, yeah. And we might want to expand on this if they're getting a little bit fancy. You'll definitely get the mark for saying enzymes denatured. But like, let's say you couldn't remember the word denatured. Oh my God, it begins with a D. Is it de-skilled? Is it degenerate? Oh gosh, what's the word? So you're going doing that in the exam. Just describe what denatured means. You could say the enzyme's active site changes, and that changes shape, and that will get you the mark. That is basically what denaturing is. Of course, you've then got to remember active site, but you might have active site in your head and not the word denatured. Then just describe what denatured is. Either of those would get you the mark. Right, let's move on. Oh my gosh, we got a graph, people. Let's get into it. There's going to be a graph in your exam, so make sure you get every single mark on your graph because 50% of the country is, and you're competing against them. So make sure you don't drop marks on the graph. Okay, complete figure one using data from table one. You should label the y-axis, use a suitable scale for the y-axis. Now, that means our scale should go up nicely and fit completely on this line, and it should go up over halfway. Plot the mean data from table one, so we're only using the mean from temperatures 25 to 45, and then draw a line of best fit, and it's worth a whopping 5 marks. So let's smash this. Oh, okay, so zooming in a little bit here, they've already put the temperature on for us. That's the first column, the independent variable. What we've got left to do here is label the units for the y-axis, and we also need to plot a scale, and that's 2 marks straight away for that. So if we look at the mean results, we are looking at the rate of photosynthesis, and what we need to do, yikes, is just make sure that it's not going over the graph. And that needs to be in, I'm putting the word in, I like to do that, centimeter cubed per hour. So there we go. No arguments there. We have scored a mark for correctly labeling the y-axis. Okay, now up here we need to do a suitable scale. I want it to go over halfway. What you're going to do is go to your results, look at the biggest rate, and the biggest rate happened at 30 degrees. It was 44 as the mean. Oh, I just used the word mean. I'm going to put mean here, guys. So it's the mean rate of photosynthesis in centimeter cube per hour. And 44 was the biggest. So I'm going to go like, I'm going to make each one of these squares 1 because that makes sense. I can easily have 44 and it will go over halfway. So there's 10. Here's 0. Here's 20. Yep. Here's 30. Getting a bit close. There's 40. And I'm going to want to go, oh my days, because 44, I can't even write properly. There's 50. There we go. And I can stop there because none of my results go above 50. And so my graph's going to take, you might say, oh, but you haven't used all of the paper. Doesn't matter. As long as it uses over half or 2/3, whatever, you're going to be sorted. And this is right there. So we're good. So let's get into it. My first result at 25 degrees is at 33.2. So nice and easy. I can come to 33, 31, 32, 33. Now it's 0.2, so I'm just going to put it somewhere just around 33 or just above 33. You might say, doesn't it have to be exact? They allow you to be one half small square out on each point. So no, you don't have to get even more accurate than that. Now we go to 30 degrees, and here is one of the biggest results, or the biggest result. It's 44. So I'm going to go up 4 above 40, and there is my result for 30 degrees. Don't know why I'm finding it incredibly hard to draw at the moment. Right. The next result is 35 and 40.8. So I'm going to go 35. Let's go 40.8. That's almost 41. So I'm just going to put it just below the line, but still above that line. And that's 35. Now to 40, 40 degrees, we drop to 22. So that's two mini squares above 20 over here. And for 45 degrees, the final result, it only goes to 2.1. So I want to climb up two squares and just go just above the line, if not just on the line. The examiner is going to give you the result for that. Now it says, draw a line of best fit. So here's the big argument. Um, Definitely 100% you will be wrong if you do this. Oh, let me do join the dots. You do not join the dots ever in any graph. But sometimes in biology, we do a straight line of best fit like this. Okay. And we would do line of best fit like that. You try to do a line going as close to most points as possible, and it's a straight line. Other times you use a natural curve, and you have to look, does it feel like a natural curve? Look, yes, it does. This is not an anomalous result. This kind of goes with what we're predicting. So this is a natural curve. You want to do one line through all the points if you can. I'm going to struggle here massively, people. I'm just letting you know. But you want to do one line through all the points without taking your pen off the paper. So is the ideal, and without any massive variations. It's quite hard on the computer when you got like wobbly hands like me. There we go. Gone through all the points. Nice straight line of best fit. Not straight line, curved line of best fit. Through all the points. One line. Don't take your hand off the paper. Um, and there we go. That's 5 marks. So how does the marking work? You get 1 mark for the axes labeling, 1 mark for the scale. See where I've gone 10, 20, 30, 40, 50. That's the second mark. We're going to get 2 marks for plotting those points. So 2 marks for the plots, and then 1 mark for the line or the curve of best fit. That's a total of 5 marks. Boom. Let's move on. Got a bad chest infection, but it's not stopping me bashing out the videos. So make sure you guys hit the like. Much appreciated. Right, question 2. Diffusion is an important process in animals and plants. What is meant by the term diffusion? This is just asking you for straightforward subject content. Now, what I recommend, if you don't know it or you can't get both points, grab a little revision card. If you don't have a revision card, get some A4 paper, just cut them into little squares to make your own revision cards. You can fold it into 8 or whatever. And write what is diffusion. Then on the back of it, write this definition. Okay. But if you know it, great. You don't need to revise it. At this point, 2 weeks out from your exams, you should only be revising things that you don't know that are going to come up. You can do a little bit of maintenance or pat yourself on the back after a hard revision session where you've gone over something difficult that you don't understand. You can revise easy stuff. It makes you feel good, more likely to revise the next day. But definitely make a note of things you don't know as you come across them in this paper and in your day-to-day revision. Right, what's meant by the term diffusion? We are talking about the movement of particles. Really, it's the net movement of particles. But movement of particles is good. That's going to get you a mark, or spreading out of particles. And it is from an area of high concentration to low concentration. Okay. Um, there we go. And that is 2 marks. Movement of particles from an area of high concentration to low concentration. You can say, if you like, a movement of particles down a concentration gradient. If you say along or across a concentration gradient, you won't get the marks. It has to be going down because that gives the impression from high to low. Okay, Coolio, let's move on. Woo! 2.2, figure 2 shows part of a leaf. Here we go. Mesophyll cell. carbon dioxide being pointed at in the cytoplasm of this mesophyll cell. We've got the stomata here, which are these two holes in the bottom of the leaf. Molecules of carbon dioxide diffuse from the air into the mesophyll cells. So they go from the air into the mesophyll cells, or from there into the mesophyll cells. Which two changes that we can make will increase the rate at which carbon dioxide diffuses into the mesophyll cells? Which two changes can we do that will increase the speed of diffusion into them? Let's have a little look. Oh, my days, slipping. Tick two boxes. Okay, so if we make less chloroplasts in the cell, that's not going to have anything to do with the CO2. If we decrease the surface area, no, that's going to reduce the rate of CO2. If we increase the CO2 in the air, yes, more CO2 in the air means it's going to be a higher concentration, and diffusion likes to go high to low, so that will speed it up. Increased number of stomata that are open, yes, that's good. So it's these two here and here. If you want CO2 to move more quickly into a cell, obviously, if you make more CO2 outside the cell, you're increasing the concentration. So you're going to get a more rapid diffusion into the cell. And if you open up more stomata at the bottom of a leaf, so there's lots more of these holes open, then more CO2 can diffuse into the leaf at a faster rate. Okay, those are the 2 marks. Let's move on. 2.3, diffusion also happens in the human lung. Whoop! Sorry about that. 2.3, diffusion also happens in the human lungs. Figure 3 shows the human breathing system. Okie dokie. Hopefully you remember what all these structures, for biology, GCSE, AQA, you need to know what all of these structures are called. Explain how the human lungs are adapted for efficient exchange of gases by diffusion. So make sure you know what these are called. This is the trachea with cartilage rings that protect it. Then we have the bronchi. Each one is individually called a bronchus. You only have 2 of them, so 2 bronchi. These are the slightly bigger tubes. One goes into each lung. Then they divide even more into bronchioles, which are these tiny small tubes which have no cartilage around them, and then they end in these structures called alveoli, which have a large surface area. And then you can see here each of these alveoli has a capillary wrapped around it. Now we need to explain how the human lungs are adapted for efficient exchange of gases by diffusion, and this one is worth 6 fat marks. So what I suggest, grab your pen, grab a scrap bit of paper, give yourself, set a timer for 6 minutes, and just give yourself 6 minutes to try and get as many marks as you can. That's what I would do. And then unpause the video and check out the answers. All right, let's get into it. So here are the points I'm going to make. I'm going to write all over this, okay, so that I can keep the diagram in focus. So this is not going to be neat, but just work with me. Okay, so the lungs have, this is my first marking point, millions of alveoli. Do you have to use the word millions? No, you can just say lots. So the lungs have lots of alveoli, or many alveoli. And how does that help? Well, it gives the lungs a large surface area. I mean, the surface is bumpy of the alveoli as well. That helps. Gives the lungs a large surface area. How does that help for rapid diffusion? So having a large surface area is going to mean things can diffuse in rapidly. So this is my first marking point, and hopefully you're going to pick up at least 1 mark for that, maybe 2 marks for that marking point there. Now, next thing, what can we say about these blood capillaries? Remember I said that there's one around each capillary. So we'd say the lungs have a good blood supply, or each alveoli has a capillary. So lungs have, you could say, a large blood supply, or extensive is even better, but I think they'll allow you to just say has a good blood supply. Lungs have a large blood supply. And how does that help? Well, that maintains the concentration gradient. I reckon this is the hardest marking point. So that maintains the concentration gradient, allowing for rapid diffusion. So it allows rapid diffusion of oxygen in to the blood cells and carbon dioxide out. And that would be my second point, although I do think this is probably the hardest point because students have to remember this difficult phrase, concentration gradient. What about concentration? Honestly, that's a bit dodgy. All right, let's go on. My spelling's always dodgy, but that word came out very weird. Right, let's go to the next one. So, what else do I want to talk about? What can you say about the capillary walls and the alveoli walls? So I'm going to say both alveoli and capillary walls are thin. How does that help? This makes a short diffusion pathway. So I reckon if you said all of that, you'd be on for 5 or 6 marks straight away, because really what you're getting the marks for is the adaptation and then how it helps. So lungs have lots of alveoli, large surface area. Lungs have a good blood supply, or a large blood supply, maintains the concentration gradient. Alveoli and capillaries have thin walls. I just wrote thin are thin. I should have wrote, oh no, I said, and capillary walls are thin. That's good enough. All right, I'll stop messing about. That will get you a mark. This makes a short diffusion distance or pathway, or a short distance for oxygen to diffuse into the blood. So that should in itself get you 5 or 6 marks. If you want, you can bus out a little bit on, they've shown you this diagram, so we could bus a little bit out on the ventilation of the lungs. We could talk a little bit about how the lungs are ventilated. So you actually don't have to know the mechanism of air in and out, but ventilation of the lungs moves air in, and this is going to maintain a steep concentration gradient. Again, moves lungs moves, let's say, oxygen rich air in, and carbon dioxide rich air out, maintaining a concentration, spell it right this time, gradient. It's actually not my spelling that's bad, it's my typing that's bad. So what do I mean by that? Listen. What am I doing when I breathe in? I'm taking oxygen in, and it means the alveoli then have oxygen close to them. So there's a high concentration of oxygen and a low concentration of oxygen in the blood. Oxygen is going to move rapidly into the blood because it goes from high to low. So we're always making the oxygen in the alveoli high by breathing in. When we breathe out, we're getting rid of that carbon dioxide. And so this ventilation of the lungs maintains a high concentration of oxygen in the alveoli, so that they're always moving from high to low into the blood. But by breathing out, we are removing the carbon dioxide and making sure there is low carbon dioxide in the alveoli, so that the carbon dioxide in the blood reaching the alveoli is higher, and so that carbon dioxide always wants to move from the blood into the alveoli so it can be breathed out. So basically, you can explain all of that by just saying it maintains a concentration gradient. And this would definitely get you a big, fat 6 marks. Boom! Right, let's move on. We're on 2.4. Figure 4 shows a root hair cell. Here we go. I always think this looks like a person's face with a really long nose, and there's the eyeball. Anywho, water molecules are shown by the black dots and nitrate ions by the crosses. Name the process by which water enters the root hair cell. So there's only one way that water moves in biological systems. You know it, begins with an O. Water moves by osmosis. So we just want to say osmosis, and that will get you the mark. Lovely. And it's going from a high concentration to a low concentration across a partially permeable membrane. Lovely jubbly. That's the whole definition of osmosis. You just needed to say osmosis. Nitrate ions need a different method. Right, if we know it's not osmosis, there's only 2 other transport methods left. Do you know what they're called? They're called diffusion and active transport. Nitrate ions need a different method of transport into the root hair cell. Explain how the nitrate ions in figure 4 are transported into the root hair cell. Use information in figure 4 in your answer. Name of the process. Justify it with an explanation for 2 more marks. Ooh, lovely. So What can we say about the X's? Are there more X's, which are nitrate ions? So are there more nitrate ions inside the cell or outside the cell? Well, there's more inside the cell. So how are we getting these X's into the cell? How are they moving into the cell if they're having to move from a low concentration to a high concentration in the cell? Well, when you move from low to high, you use active transport. And so that is what we need to put down as our answer. Let's go. So, name of the process is active transport. Explanation: nitrate ions are moving from a low concentration where outside the cell or in the soil to a higher concentration inside the cell. Diffusion would actually be taking the nitrate ions out, which is a bad thing. So from high to low. So active transport has got to overcome that. And there we go. And why do we need, what else do you know about active transport? The word active, think of the word active. What does active mean? It means movement, it means energy, and they use energy. So we're going to say using energy from um, well, it doesn't have to necessarily be aerobic, but we're just going to put respiration. It could be aerobic respiration, normally is aerobic respiration. So active transport, nitrate ions move from high to low, sorry, low to high, using energy from respiration. And that is 3 marks. How did you do? Okay, guys, we're moving on to question 3 out of 7. Woo! Figure 5 shows 3 types of cells. We've got a bacterial cell, which is 1 micrometer wide. We've got a liver cell, 30 micrometers wide. And we've got a mesophyll cell, which is 0.05 millimeters wide. Oh, you cheeky little bums. What I'm going to do here is I'm just going to highlight that because look, the units are different. So we're going to need to take that into account. Give 2 similarities between the prokaryotic cell and the eukaryotic cells in figure 5. Okay, so we don't need to talk about this business here because we're talking about similarities, and these sizes are all not similar: 1, 30 and 0.05 millimeter. Right, so what are 2 similarities between the prokaryotic cell, which is this one, and the liver and mesophyll cell? These guys, by the way, are eukaryotic. Why? The mesophyll is from a plant and the liver cell was from an animal, and both of those have a nucleus, so they're both eukaryotic. Okay, what are the 2 similarities? Let's name something they all have. Can you see these black dots? What are those black dots? Those are ribosomes. So we can say they all have ribosomes or ribosomes. What else do they all have? Well, they don't all have mitochondria. Look, these two do, but that one doesn't. What's this gray stuff that fills both the cells, the background color in the middle? That is the cytoplasm. So I would say they all have cytoplasm. And also, what's this black line that we draw around the outside holding the cytoplasm in? Look, that's got it. That's got it. That's the cell membrane. So I'd put that as another one. They all have cell membranes, and you can be guaranteed that these are things that all cells have: ribosome, cytoplasm, and cell membrane. So they're always your similarities in cells. Yes, they all have DNA. You can't really see that from this. The DNA is inside a nucleus in the liver and mesophyll cell, and this has DNA floating in the cytoplasm. So we could have said they all have DNA. That's also a possible answer. Right, let's zing on, folks. Give 3 differences between the prokaryotic and eukaryotic cells. Right, I need to go up so we can see the actual diagram. That'd be nice. Right, I'm going for 3 differences now, guys. So I'm going to write down 3 differences up here, not where the question is. So you might want to pause the video, study this diagram, write down 3 things you can see that are different, and then unpause and check your answers. Okay, let's go for it. Right, well, bacterial cell has no mitochondria. So bacterial cell um, or actually, yeah, has no mitochondria. The cell, it was probably in context of prokaryotic, so I should really say prokaryotic cell was no mitochondria, mesophyll and liver cell do. I want to say as well, bacterial cell has no nucleus. Okay, so that's two differences. I can't say bacterial cell has a cell wall because so does the plant cell. But what I can say about the cell wall, check this point, bacterial cell wall is not made of cellulose. You don't actually have to know. It's made of something called peptidoglycan. Uh, peptidoglycan, but you can call it murian. It's made of different substances. You don't need to know that, so don't worry about it. All you need to say is that the bacterial cell wall is not made of cellulose. That will get you the third mark. But there's other points we can make as well. The DNA in the bacterial cell is in the cytoplasm. So the DNA is found in the cytoplasm. I could also say the DNA is circular, whereas the DNA in the liver and the mesophyll cell are most probably double helix, if you get me. Um, what else? What else? Yes. So I could put that as well. This is in regards to the bacterial cell. The DNA in bacterial cell is, I swear I'm going on a bit long here, longing this one out. DNA is circular. What else? What else? They actually have smaller ribosomes. Oh, I could also say it is smaller. So bacterial cell is smaller. Now, I probably shouldn't be using the word bacterial because the question was asking us to call it a prokaryote. But there we go. I put five differences. Okay, five big bad boy differences. Right, let's move on. Let's move on. Um, so you needed three of these. There's probably more. You can check the mark scheme if you like for that. Right, let's zing, zang, zong. 3.3, calculate the ratio of the size of the bacterial cell to the mesophyll cell. So we want to do the ratio of the two cells. So the ratio is 1 for the bacterial cell. What is the mesophyll cell? Uh, Generally, plant cells are eukaryotic cells. They're going to be quite a bit bigger. First thing I need to do is study this. Say that's in micrometers, that's in millimeters. You can do change either one. It's up to you which one you want to change. So you either change this to millimeters or you change this to millimeters. So change this one to micrometers, sorry, or this to millimeters. If I want to change this one micrometer into millimeters, I'm going to divide by 1,000. Now this screws with people's heads, but when you're going up the scale, you divide by 1,000 for each step in the scale. So if I want to go from nanometers to micrometers, I divide by 1,000. If I want to go from micrometers to millimeters, I divide by 1,000. If you wanted to convert millimeters to meters, divide by 1,000. So it's kind of counterintuitive, but as you go up to bigger units, you divide by 1,000. So 1 divided by 1,000, what are we going to get, folks? What is that? 0.001? I believe so. Let's check it out on the calculator just so I don't make a huge floppage. Yeah, 0.001. So equals 0.001 millimeter. I've now got them both in millimeters. That's the bacterial cell. How many times bigger is the mesophyll cell? Well, I'm going to do 0.05 and I'm going to divide it by 0.001. Or if you don't like that, just divide them both by 0.001. And that's going to give me 1 here in my ratio. And what's that? 50? Let's check it out. 0.05 divided by 0.001 equals 50. Yeah. So the answer of the ratio question is it's 1 to 50. So the mesophyll cell is 50 times bigger than the bacterial cell. I could have done this by converting to both to micrometers. In that case, if you want to see what I do if I convert it to micrometers, I'm going to times that by 1,000. And then this is the other way to do it. If I times that by 1,000, that's going to give me 50 micrometers. And then we know that the bacterial cell is 1 micrometer. So we get the same answer, 1 to 50. Okay, either of them is good, peeps. That'll get you all the marks. 1 to 50. Okay, name the type of cell division that produces genetically identical body cells for growth and repair. So I like to think of the answer, of course, is mitosis. How do I remember it? I say mitosis happens in mitoses. Okay, so it happens all over your body, everywhere, and it's for growth and repair. Meiosis, if we check meiosis, this is the other type of cell division. Meiosis, I like to think of the E for egg and the S for sperm. So meiosis only happens when making eggs or sperm. Mitosis happens in your toesies. It actually happens everywhere. All right, let's go, let's go, let's go. That'll get you a mark two. Let's move on. Figure six, I've got a bad chest. So if you hear me go like a lunatic, that's because my chest has been busted for the last 3 weeks. Figure six shows a cell cycle. Okay, we've got stage one, stage two, stage three. There are three parts to the cell cycle. What percentage of time, what percentage of the time for one cell cycle is represented by stage two and three? So this is just how to use a pie chart, basically. That's 50%. They've broken this up into 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. They've broken it up into 20. So you're looking at 1, 2, 3, 4, 5, 6, 7. So we're looking at 7 divided by 20 times 100, and that is going to give us 35%. Okay. 35% is represented by stage two and three. These bad boys are 35%. Okay, let's zing down. So the correct answer, folks, was this one, which was 65%, stage one. Um, Chromosomes, or you can say DNA, or you can say genetic information, and you can probably say genes. Jeez Louise, that is terrible. Doubles, you could use the word replicates, you could use the word reproduces. So in that first stage, the DNA and the chromosomes or the genetic information double. Um, Specifically, actually, in AQA, they're being really poncy and they don't want you to say the DNA or genetic information. They want you to say the chromosomes reproduce. Wow, they are being fancy wancy. That's one mark. Right. Now, second mark, new cell structures are made. So we could just say new cell structures are made. And what do we mean by that? You could give an example, more ribosomes or more mitochondria. You could say more cytoplasm, whatever. Obviously, if you're going to divide, you need to make more bits before you divide. Now, in stage two, you can say anything from mitosis. So stage two is mitosis. Now, they don't give you a mark for the name. Mitosis, what happens? You might remember these are some of the things you could say. Chromosomes form X shapes, line up on middle of cell. So they line up on what we call the equator, the middle of the cell. Um, X shapes pulled apart. Each arm goes to a different end of the cell. So there's a bunch of things you can say that would describe mitosis, but essentially mitosis is when this happens. Mitosis is when the chromosomes form like X shapes. They're kind of random at first, like this, but then they line up in the middle of the cell like that. And then the chromosomes pull apart. So you could have described any of this. They then pull apart the different arms, going to different ends of the cell. And now the final stage is called cytokinesis. Do you need to know that? No, you don't. Cytokinesis means kinetic, like moving. So the cytoplasm moves, the cytoplasm divides. So we want to say cytoplasm divides, or we can say cell splits, divides into two daughter cells or two new cells. And there we go. I think that was a difficult question. So if I was you, I'd make a revision card on that because a lot of people won't know the different stages of the cell cycle. And it's made up of three things. Basically, the cell has to double all the stuff inside it. So double the ribosomes, double the mitochondria, double the cytoplasm, etc. It needs to double its chromosomes. Each chromosome contains a DNA molecule bound to a protein. But why you can't say DNA is beyond me and is stupid for stage one. You should be able to say it doubles its DNA. Anywho, stage two is the mitosis bit where the chromosomes get ready to kind of like move to each end of the cell. So they line up on the middle of the cell and then they split apart, half the chromosomes going to one end, half going to the other. And then stage three, cytokinesis, is when the cytoplasm splits. It literally, the cytoplasm buds like that and then tears. First of all, it kind of buds, then it tears off, and you end up with two new cells that are identical to the original parent cell, each of those cells. Anyway, let's move on to question 4. Hope you're doing well. Don't forget to hit the like, folks. Last couple of bits. Describe what happens during each stage of the cell cycle. So the lipases break down lipids. Which two products are formed when lipids are broken down? So you need to know this off by heart. Amino acids come from proteins. Um, Lipids are broken down into fatty acids. Glucose, no, that's a monomer of starch or glycogen. So, and fatty acids and glycerol make up lipids. So if you don't know that, put it on a revision card because it's a difficult one because biology has a lot of G's. Glycogen is actually a polymer of the monomer glucose. It's like starch, but heavily branched. Um, so the correct answers here, I'm going to get off red because red's a disgusting color to use. Um, yeah, so the answer is acids and glycerol. Remember, there are 4 Gs in biology. There's glucose, which is a monomer, a sugar, simple carbohydrate. You have glycerol, which is a small molecule found in lipids. And you have glycogen, which is a polymer of glucose. And you also have a hormone that comes up in paper 2 called glucagon that lowers the blood that increases the blood sugar level when the blood sugar level or the blood glucose level is low. So, and it's found in the pancreas. So you might want to make a revision card about the 4 Gs. So that's glucose, monomer, simple sugar, glycerol, something that makes up lipids, glycogen, which is a polymer of glucose found in the muscle cells of animals and the liver of animals. And we have glucagon, which is a hormone found in the pancreas, and it is for the control of blood sugar. Okay, coolio, let's move on. One model used to explain enzyme action is called the lock and key theory. Figure 7 shows the model of the theory. We have an enzyme, we have a substrate, they join together, and then afterwards we have products. Explain the lock and key theory of enzyme action. Use the information from figure 7 in your answer. So first of all, we want to say the substrate. Do you want to say it has the same shape? No, that shape is not the same. They're completely different shapes. However, they do fit together like a jigsaw puzzle. So we use the word complementary. But if you can't use that word, just say the substrate fits in the enzyme like a lock in a key. Nice, right? So I'm going to say substrate has a specific shape that is complementary to the shape of the enzyme's active site. They fit together like a lock and key. Boom! So, what are the marks for here? The marks are for saying that the substrate has a special shape and it's complementary or fits perfectly into the active site. They fit like a lock and key. We actually call this in A level an enzyme substrate complex once they're joined together. And then what it does is it weakens the bonds. Lowering the activation energy. If you think of your energy profile diagrams in chemistry, it lowers that activation energy and products are released at the end of it. And at the end of it, the shape of the enzyme is not changed. It's ready to go again. It's a catalyst. So I can also add here that the substrate is changed into products and the enzyme remains unchanged. It is a catalyst. So it can be used again afterwards. Okay, folks, let's move on. There are many different types of lipase in the human body. Why does each different type of lipase only act on one specific type of lipid molecule? So what they're saying is there's lots of different fats called lipids in our body, and each different type of fat that you could eat, like you might eat fat from some sunflower seeds, or you might eat fat from some olive oil, or fat from that's found in a bag of chips, or fat from a cheeseburger. Each of the different fats needs a different enzyme to break it down. Why? And the idea is it's really simple. Um, each lipid molecule has a different shape. I'm going to put a fancy answer, so it has a different 3D shape. So the active site of each lipase enzyme has to be complementary to that specific molecule. So it's basically different lipids have different shapes. You get the idea. And the active site has to have a special shape that fits a specific lipid molecule. It can't fit all of them. Okay, quite a difficult question. Students investigated the presence of starch and glucose in the leaves of geranium plants. This is the method used. Place two identical geranium plants on a bench near a sunny window for 2 days. Ah. After 2 days, leave one plant near the window for 2 more days. Lucky plant. Place one plant in a cupboard with no light for 2 more days. Bit of a Harry Potter moment there. Remove one leaf from each plant. Crush each leaf to extract the liquid from the cells. Test the liquid from each leaf for glucose and for starch. Describe how the students would find out if the liquid from the leaf contained glucose. So this is basically a long way of asking you, how do you test for sugar? And you should know this off by heart. If you don't, grab a revision card, write it down. It's 3 marks. Use Benedict's reagent. You can just say do the Benedict's test. They just want to see the word Benedict. Okay. So you're going to warm. You're going to crush the leaf, then you're going to warm or heat um, if it contains sugar stroke glucose, it will turn red. And there you go. 3 marks for the Benedict's test, peeps. You just need to know Benedict's test is for sugar, warm it, turns red. I should say that yeah, the marks are for Benedict's reagent, heat, and turns red. You can say turns green, turns yellow, turns orange, or red, but with most, yeah, that's kind of semi-quantitative, so it gives you an idea of how much there is. Red means more sugar than orange. Orange means more than yellow, and yellow means more than green. Green is just a slight bit of sugar. All right, that's your Benedict's test. Describe how the students would find out if the liquid from the leaf contains starch. So they're testing you on the old starch one. I mean, it's a little bit more complicated than this, but in AQA, you only need to simplify it. What is the chemical that we use to test for starch, and what is the color change? And that is all you need to say. So we want to say add iodine solution. Iodine is actually a black solid, so you have to dissolve it to make that kind of brown orange solution. It will turn blue black with starch. It starts off orange. Now, do you get the marks for saying what it starts off from? You can just say it changes to blue black. You don't need to say it was orange to start with, but it's good if you know what color it is to start with. I mean, really, it wouldn't really work very well on a leaf unless you boil the leaf with alcohol and remove the chlorophyll, because then you can't see a color change with chlorophyll in the leaf. But you don't need to know that for AQA. Test glucose starch leaf from plant kept in light for 4 days. So when the plant was in the light for 4 days, there was lots of sugar, a strong glucose test. Leaf was kept in the light for 2 days and then no light for 2 days. So it was Harry Pottered in the dark for 2 days. It only had a weak glucose test. Starch, 4 days in the light, really strong positive starch test. However, when it was kept in the dark, negative, no starch. Explain why the leaf in the light for 4 days contained both glucose and starch. So what we want to say here is um, it used the light to photosynthesize, and that will get us a mark, right? It's using the light for photosynthesis. What does photosynthesis make? Um, Photosynthesis makes glucose, makes sugar, right? So we're going to get a mark for that. And then what else do we want to say? Where does the starch come from? Well, glucose is turned into starch. So then some of the glucose, this is called assimilation. Some of the glucose is turned into starch. You actually have to know the 5 uses of glucose in plants for paper one. And the 5 uses of glucose in plants are turning it into starch, using it for aerobic respiration, using it to make lipids, using it to make proteins, and using it, you could say, to make chlorophyll. I think there's some other uses as well that I haven't mentioned, but there we go. Right, so one of those is that. So there we go. Ooh, missed the question, Tom. Explain why the leaf in the cupboard with no light did contain glucose but no starch. So um, this is kind of like, first of all, the plant's going to use the glucose. Remember, we said it had glucose and starch. So it's going to use some of the glucose up to respire. So used glucose up to respire. And that might be 2 marks for us because we're saying it's using the glucose for respiration to release energy to stay alive. Um, so use glucose up to respire. I might want to say to release energy because it's trying to stay alive. Now, what's it going to do with the starch? Starch is a polymer of glucose. So starch is a polymer of glucose. Um, the plant broke down the starch into glucose so it could use that for respiration too. It might have wanted to use that glucose to make proteins. It might have wanted to use that glucose for other things, but essentially it needed to use that glucose for like metabolic reactions, so to like make stuff. Um, so it had to break up the starch. The starch is a long-term energy store. So using up the glucose for energy, for respiration, starch is a polymer of glucose. It got broken down. Okay, cool. Right. Suggest one way the students can develop the investigation to find out more about glucose and starch production in plants. So let's go up and with this kind of suggest you want to look at what they did do and then suggest something else. So they tested leaves. Here we go. Here's the method. The students investigate the presence of starch and glucose in the leaves of geranium plants. So how about maybe they could have tested the stems and the roots. They did them in the light and they did them in the dark. Um, maybe they could leave them longer in the light or leave them longer in the dark. Maybe they could do other plants because look, they use geranium. That's not all plants. So cool. We can add that as suggestions. So you normally find the answers to the suggestions hidden in the method. So use different species of plants, not just geraniums. Okay. So what happens when I type things go wrong, huh? We could have also said test the leaf, the roots or stems. You get the idea. So different things they could have done. All right, let's move on. Oh my gosh, question 5. Many plants have evolved defense mechanisms. Figure 8 shows a gorse plant and a part of a deadly nightshade plant. Gorse, deadly nightshade. Gorse plant has evolved to have sharp thorns. So we can see there, that's going to give you a nasty little prickle if you bite that. Um, Whereas the deadly nightshade, I guess them berries are pretty deadly, hence the name. What type of defense response are the thorns? Right, there are three defenses you guys have to know. Okay, so there is chemical defense, there is physical or mechanical defense, and then there is physiological defense. So, which one is this? This is a mechanical defense or structural defense. So it's the anatomy has changed. And when you bite that, ow, it's going to hurt. So I call this a structural or mechanical defense. Um, and how does that defend the plant? Herbivores um, get spiked. Ouchie! Yeah, stops animals. I'm going to call them herbivores. They're the ones that eat plants, goats, rabbits, things. Stops herbivores eating the gorse plant. Okay. Oh! The deadly nightshade has poisonous berries. What type of defense is poison? Poison is a chemical. Okay, so it's a chemical defense. So obviously that works by when the rabbit or whatever herbivore it is eats a few of the berries, it gets sick, starts to vomit, and yeah, it goes, I'm not eating from that plant again. 5.4. A scientist noticed that in one area, the gorse plants had yellow leaves and stunted growth. Stunted means shorter, in case you're wondering. One reason for yellow leaves and stunted growth is a deficiency of nitrate ions in the soil. So if there's not enough nitrate, you get yellow leaves, you get stunted growth. Explain two other possible reasons for the yellow leaves and the stunted growth. Do not refer to nitrate ions in your answer. So there are other minerals that are needed, and one of them is magnesium. And magnesium is needed. Oh, what I need to say, lack of magnesium in soil or in the plant. Magnesium is needed to make chlorophyll. Um, chlorophyll is green in color. Without chlorophyll, plant cannot photosynthesize um, to make glucose for respiration, for energy, for growth. Oh my days, that's long. Yeah, and also if it wants to make proteins, it needs the glucose to add to the nitrate ions to make proteins. So anywho, there's my big fat explanation for that, and that is going to get you definitely 3 marks. Reason number two, I'm kind of like going to have to write my explanation here as well, guys. So I'm going to like squeeze it onto the page. So another reason we could go for, it's got diseased. We could name a disease. What are two plant diseases you know of? You got rose black spot, you got tobacco mosaic virus. So I'm just going to say it has tobacco mosaic virus or TMV, which discolors leaves by destroying chlorophyll. And again, the same reason, it cannot photosynthesize. And then I could go down the whole route above. So cannot make glucose, less respiration, less energy, or less proteins can be made. So go down that route. Less protein, less growth, less repair. Okay, I'm running out of room. Cool. Right, the gorse plant has nodules on its roots. So these are the roots, and these little balls are the nodules. The nodules are part of the living tissue. Bacteria which convert nitrogen gas into nitrate ions live in the nodule tissue. So there's bacteria living there that take nitrogen gas from the air and convert it into nitrate ions. Suggest how the nodules benefit the bacteria. So they're really asking, why would bacteria want to live in these little balls? So it basically protects them from predators. It's a place to live. Maybe they can feed off the plant, and maybe the plant provides glucose. They can use some of the glucose the plant makes in photosynthesis. So I'm going to put that. So I'm going to put bacteria get shelter or protection by the nodules. Yeah, so they're getting protected by living in the nodules. Um, Also, they get glucose from the plant that they can use in respiration, um, for making substances or releasing energy. So all sorts, all sorts, all sorts. I'd probably go with these more than the shelter. There aren't specific predators maybe being put off by the nodules, but yeah, thought I'd give it a go. There we go. Right. Explain how the nodules benefit the plant. So why does having bacteria living with them benefit the plant? Well, bacteria convert nitrogen into nitrates. The plant can absorb the nitrates and use them with glucose to make proteins. And what do you know about proteins? Proteins are used for growth and repair. So basically, nitrates are used with glucose to make amino acids. I could have long this. I could have said bacteria convert nitrogen into nitrates. Bosh, Mark. Plants can absorb the nitrates and use them with glucose. I could say to make amino acids, then to make proteins. Proteins are used for growth and repair. That's a long explanation, but there's plenty of marks there. For many years, drugs have been extracted from plants. Which plant was chewed as a painkiller? Tick one box, one mark. Okay, blackcurrant berries, no. Foxglove leaves, no. Now, foxglove, you do need to know. So if you don't know, make a note of it, put it on a revision card. Foxgloves are where we get the drug digitalis from, and that is something that slows down your heartbeat or corrects the rhythm of your heart. Don't ever go and eat a foxglove. They're actually really dangerous and can kill you. Rose petals, no. Willow bark. So aspirin was first found in willow bark, the willow tree. So the bark of the willow tree was made into a tea in China thousands of years ago to as a painkiller, and maybe for some other benefits. But yeah, that's where we got aspirin from, the willow bark. So there you go. Right, let's go on to question 6. I believe there's only 2 questions left, folks. So let's get into them and get through them as quick as possible, because this one is taking time. 6, data from Million Women survey in the UK was collected for over 15 years. Scientists analyzed data to study the effect of consuming alcohol on liver disease. Scientists included 400,000 women who regularly drank alcohol, included 400,000 women who did not consume any alcohol, excluded women who already had liver disease. Age and gender were 2 factors they controlled in the analysis. Many other factors were also controlled. Suggest 2 other factors the scientists would have probably controlled here, guys. So we're looking at liver disease, so and anything to do with your health. So I'd say I'd want them to have a similar diet. I can never spell that. I hate that. Smoker or non-smoker is probably something that might play a factor, right? We should control whether they're smokers. What medicines they take, like for instance, I'm an asthmatic. I take inhalers or use those like puffers. So obviously that might have an effect on your liver. What about genetics? Well, family history, so of liver disease could be one of the things we want to go with. Oh my days. So similar, what did we go for? Similar diet. Well, sorry, my charger ran out there. I just had to run and get a charger. What medicines they take? Where were we? Where were we? Family history. Yes. What's that about? Well, has anyone else in your family had liver disease? You might not have it, but has anyone had it? So that might affect the results of the experiment. We might also look at fitness levels, which means like how often, how sporty they are, how often do you go to the gym? What about ethnicity? Ethnicity. So yeah, ethnicity as well. So maybe liver disease is more prevalent in different ethnicities. And that might be a factor that we want to control as well. Okay, so yeah, let's move on. Data was analyzed for women who drank alcohol with meals, and then they analyzed women who drank alcohol not with meals. Women who did not drink alcohol at all. During the survey, approximately 1,500 women developed a liver disease called cirrhosis of the liver. Scientists calculated the relative risk of developing cirrhosis of the liver for each group who consumed alcohol. A relative risk of one means there was no statistical difference between the group who had the alcohol and the ones who didn't drink any alcohol. So they use the people who didn't drink alcohol as like the baseline, right? What their risk is of getting liver disease. You can still get liver disease if you don't drink alcohol. So they were using that as like the baseline and then comparing the people who did drink alcohol with that. Okay, risk of developing cirrhosis of the liver. We got two lines, black line, dotted line. I guess the black line, the solid black line is going to be the alcohol drinkers, right? Yeah, consumed alcohol, but not with meals. Oh, consumed alcohol with meals. So actually, sorry, both these lines are for people who drunk alcohol. Number one on there is the risk of you getting the disease if you don't drink alcohol. So number one, there is a risk, but number one represents the risk of you getting liver disease, cirrhosis, even if you don't drink alcohol. So you can see the people who drunk alcohol without eating meals at the same time were at a greater risk, it looks like. A woman drinks 150 grams of alcohol per week, not with meals. Ooh. The woman decides to change to drinking 150 grams of alcohol per week with meals. Calculate the percentage decrease in the relative risk of developing cirrhosis of the liver for this woman. So, what we want to do when you're doing percentage change or percentage decrease, you want to do the difference over the original. So, what was her risk at first? Well, she drank 150. So, we want to look what was her risk at first. 150 grams, we draw a line up here and then we draw a line over there. She had a risk of 2.5. So, I'm just going to put that over here. Her original risk was 2.5. Then, what was her risk after um she started to say, nah, I'm having meals with it, boy. And there she started to have meals with her alcohol and eat whenever she drank. And her risk went from 2.5 to this. So they're testing your reading of a graph. Each one of these is worth one because there's 10 of them between one and two. So 1.6 her risk went down to. So the difference is 2.5 divided by 1.6. That's the difference. Divided by the original, which was 2.5. And then what we want to do is times 100. So 2.5 take away 1.6 is 0.9. So essentially we're doing 0.9 over 2.5, change it into a percentage by timesing by 100, and we are going to get like 37.2, I don't know, 549, something like that. It's up to you what you round that to. Did they want it to a whole number? Well, we can just leave it with percentage as 37%. Or if you want to round it to one decimal place, I'd go like 37.3%, something like that. So, yeah, there we go. Let's move on. There was a 37% decrease in her risk, so well done to her. One glass of wine contains 12 grams of alcohol. A different woman drinks two glasses, women, a different woman drinks two glasses of wine each day with her meals. Calculate the relative risk of developing cirrhosis of the liver for this woman. So she's drinking two glasses, that's 24 grams with meals. What is her risk? Oh, this is units of alcohol per week, folks. We need to work out how much she's drinking per week. So she's drinking one glass. She's drinking two glasses a day. So we want to go 2 times 12. She is having 24 g a day, 24 gram a day. We need to times that by 7, which what does that make? 148. Am I right? Am I right? No, 168, you plonker. Right. So she has 168 grams per week. Now to calculate her, she has it with meals. Bosh. We want to go to the graph, find 168, draw a line up, go across, read off her relative risk. Let's do it, folks. So she has 168. So I'm going up here 150. Buh, 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 buh. how much is each one of these? Each one of these is worth, oh my good gosh. Each one of these is worth 5. Each mini square is worth 5. So that's 160, 165, 168 is about, that's 175, 170. It's about here. My days. My stomach is gurgling. So 1.8 is the answer there, folks. So that is the answer. And that will get you 2 marks, one for the 168, one for the 1.8. Let's take this down and show you where it should go. So her relative risk is gonna be 1.8. Yeah, quite a difficult question. You had to use the graph. You had to make sure you circle and underline things in questions. It's really important. Get into the habit of being active with your exam, actually getting involved with the exam, underlining keywords, circling. It doesn't mean you have to use those key things or that they're relevant, but you're more likely probably to pick up and not miss something blatant. People miss so many marks that they're like, oh, I could have got that. Okay, we've had this repeated. Consuming alcohol with meals instead of not with meals decreases the risk of developing cirrhosis. Yeah, we can see that. Give 2 other conclusions about the relative risk of developing cirrhosis of the liver related to alcohol consumption. We got to use data from figure 10 in our answer. Yeah, yeah. Okay, let's check it out, peeps. So what we can say here, look, if you drink alcohol, your risk is increased. And whether it's with meals, without meals, or with meals, it's increased relative to people who don't drink. Okay. And we can give evidence for that. We can say, look, we can just give any data. So we could say, look, if you have 100 grams of alcohol a week with a meal, your risk goes up to 1.2. If you have 100 grams of alcohol a week without a meal, your risk goes up to 1.65. And so there you go. Just pulled out some data from the graph and smashed that into the answer. Okay. So I'd use supporting data to back that up. Well, one thing we can say is, look here, look at this section. It's a straight line there. So actually, if you drink less than 50 grams of alcohol with a meal, your risk is the same as someone who doesn't drink alcohol. It has a risk of 1. So actually, as long as you don't have more than 50 grams a week and you have a meal, the evidence is showing that you're not more likely to get cirrhosis, damage to your liver, than someone who drinks no alcohol at all. So actually, 50 grams of alcohol a week is perfectly acceptable in terms of liver damage. Whoa, where am I? Oh, here I am. So I would say drinking less than 50 G a week with a meal gives a risk of 1, which is the same as not drinking alcohol. And then I'd also say both drinking alcohol with and without meals increases the risk of liver disease. Now, I can't be bothered, but I'd back that up with evidence from the graph. So use some data from the graph to show that last statement, and that will get you the marks. So you can just say like 100, whatever, the risk is this, is 1.2, I believe, at 100 grams of alcohol. It was 1.2 with meals, and at 100 grams without meals, it was 1.65. So I'm going to put that down there anyway as an answer. So at 100 grams with meals, risk 1.2, and 100 gram without meals, risk 1.65. The examiners love a bit of data to show that you know how to use a graph. Suggest 2 reasons why the data is valid. So this means to have valid data that people can say, yeah, this is true. They must have done some good things in their method, things they must have controlled or done that are really respectable in science methods. So let's go up and check out their science method. What did they do that was good? Let's circle the things that you go, yeah, that's a good experiment. They analyze women who drank alcohol with meals, not with meals. They did the survey. 1,500 women developed liver disease. Scientists. Um, hold on. I've not even looked at the method. The method's up here somewhere. Here we go, folks. Scientists analyzed the data to the effect of. Oh, it was collected over 15 years. That's pretty good, isn't it? They've done the study over 15 years. So the data is very reliable. Also, 400,000 women they used, a total of 800,000. So they used a really large sample. That is really good. And they didn't let anyone who's already got liver disease take part. So I think these are the things that make it a very valid, um, the results valid. And they controlled for age and gender. We can say that as well. They controlled for age and gender. So let's go down. Uh, they did study over a long time, 15 years. Um, they used very large sample sizes of 400, whoa, 400,000. So they used 400,000, 800,000 in total as their sample size. Lovely. Right. Let's zing. Suggest one aspect of the survey which might reduce the validity. So what does that mean? That means something that they didn't necessarily control for that actually might make it less reliable. Um, so people might have lied about how much they drink. So it relied on the honesty, way honesty of people when finding out how much they drank. So, for instance, some people might say, "Oh, I only have 2 glasses a week," and yet they're guzzling the old bottle down. So that could kind of interfere because it was survey results. Nobody actually watched them do it. Also, maybe the spacing of the drinking. So, like some people might drink their alcohol 1 glass a day. Other people might just have 10 glasses of wine on a Friday night and then don't drink for the rest of the week. We're not sure how that affects the liver because they didn't take account of the spacing of the drinking. Right, cirrhosis of the liver leads to liver failure. Describe the effects of liver failure on the human body. So this is a really difficult question. It's called an application question. And actually, what they're really testing you on is, do you know what the liver does? So you might be sitting there going, oh, we've never done liver failure in school. This is outrageous. Just talk about the things you know that the liver does. So you should know the liver makes bile. Without bile, let's just say if it can't make bile anymore, fats will not be emulsified, right? So if you can't emulsify fats, less absorption, so less energy. Maybe you've got less energy, less insulation. What do we need fats for? We need fats for insulation. We need fats for energy. So if you can't absorb as much fat, you're not getting that fat in your diet, right? What else does bile do? Bile also neutralizes um, stomach acid. Um, without this, the stomach acid may denature other enzymes like amylase or lipase, and therefore um, you do not absorb as much glucose for respiration. There you go. Look, I'm whacking in as much waffle as you can. So you can really go to town with this one. Also, the stomach acid may damage your villi, right? You're not supposed to have stomach acid in your intestines. Stomach acid may damage villi, decrease surface area, and then you could go into a whole spiel about decreasing the surface area reduces your ability to absorb nutrients like amino acids for making proteins for growth of muscle. There's like a whole heap of things you could go on with here. Also, what else does the liver do? Because I've just been talking about bile, but the liver, of course, the liver also is where lactic acid is broken down. We learned that in unit 4 of paper 1. in the bioenergetics unit. So the liver is where lactic acid is broken down. If you get too much lactic acid, muscles will be sore and painful. So you might be in a lot of pain if your liver doesn't work because you can't break down lactic acid. So lactic acid builds up and your joints and your muscles are really painful, and that could be really bad. And of course, the liver also detoxifies the body, but the liver is also a store of glycogen. We learned that in paper two, which is more of a paper two thing, but liver is also where we learn that when we're doing the control of blood sugar. Liver is also where glycogen is stored. Um, yeah, um, plays a role in regulation of blood sugar levels or blood glucose levels. And then if I want, I could go into a long one about how without the control of blood sugar, you might fall into a coma. You might develop diabetes, something like that. So we could go on and on and on and on. Anywho, I'm going to move on to the final question because this paper has taken too long. Monoclonal antibodies, MABs, MABs, are usually made from mouse lymphocytes. Candida albicans infection produces serious symptoms in patients with a poor immune system. Recently, scientists have produced MABs to Candida albicans using human lymphocytes produced naturally after an infection. Candida albicans lives in the throat of infected patients. The sample is taken from the throat of a patient with suspected Candida albicans infection. The sample is transferred onto a microscope slide. Describe how the MABs and fluorescent dye could be used to see any Candida albicans pathogen on the slide. Oh my days, that was a serious stem to that question. So I imagine that the fluorescent dye binds to the monoclonal antibodies, right? And then what you can do, you can wash it with water, see if there's any left, and then it will show up under a microscope and you'll be able to identify um, the pathogens Candida albicans. So, yeah. So, fluorescent dye binds to the monoclonal antibodies. We're just calling them MABs, although I don't know how to. Yeah, yeah. So, the fluorescent dye binds to the monoclonal antibodies. Then put these on the slide. Um, they will bind to the Candida um, albicans, or what was it, a bacteria. They're going to bind to the bacteria, identifying under the microscope the presence of the bacteria. So yeah, you would actually wash it afterwards. So you would add the dye and the MABs to the slide. The MABs and the dye are already bound together, like glued together. If the MABs join to the bacteria, then when you wash the slide, they will still be there and they will show up under the microscope. Okay, that's all long Maximus. Very difficult question that, so don't punch yourself in the face if you didn't get any of that right. In the laboratory, the human lymphocyte MABs were injected into animals infected with the Candida albicans. The MABs caused the increase in phagocytosis of Candida albicans pathogens. Doctors intend to start a trial to give the MABs to patients severely ill with Candida albicans. Explain how the increased phagocytosis of Candida albicans pathogen will help the patient. So, what does phagocytosis do? Right, it engulfs the pathogens, digesting them, um, so they cannot produce their toxins, right? That damage cells. And that is the route that I am going down. Less toxins produced, less cellular damage. Okay, I think. Oh my gosh, 7 more marks. Jeez, we're nearly there. 7.3. It's been shown that MABS treatment is effective in the laboratory using both infected tissue culture cells and infected live animal cells. MABS treatment for Candida albicans is now ready for clinical trials on people. Describe how the clinical trial should be carried out. So, just to refresh your brains, I would say grab some paper, grab a pen, pause the video, have a go. You're talking about the clinical trials, the trials on people, not the trials on animal tissue and animals that are carried out. They're called preclinical trials. So let's describe how they should be carried out. So first of all, I want to say first stage, give to healthy volunteers. Why do we give it to healthy? Actually, it doesn't say explain. Ooh, check that. It's describe. So we only have to say what happens, not explain it. So first stage, give to healthy volunteers. If you wanted to know why they do that, it's in case there are any side effects, and sick people with weaker immune systems, the side effects would be worse for them. So anywho, we give to healthy volunteers in the first stage. Um, this is to test for toxicity and side effects. So we want to see if there's any side effects or if it's toxic or dangerous. Okay. We also then we test on patients to find the optimum dosage. So we want to find the dose where the patients get better, but they have the minimal side effects. We call that the optimum dosage. Okay. Also to check what dose gives less side effects. To be honest, that's what the optimum dosage is. The dose that gives less side effects um, and still um, is effective. Okay, so we want it to the where its efficacy is at the best with the least side effects. That's the optimum dosage. Right now, how should you carry this trial out? They should do a double blind trial where the doctor and patient are unaware who is taking placebo and who is taking the medicine, okay, or the drug. And that should get us all the 6 marks. But what you could also add is use a large sample size and use repeats and calculate means. Repeats help you spot for anomalies, so we could have put that as well. That's going to get you all the fat 6 marks. Right, last part to this test. I'll be glad to get this done. Scientists have also used human. If you can hear barking in the background, my crazy little chihuahua's in the garden at the moment. There's just been barking for the last half an hour, so I'm going to go punch it up in a minute. Right, 7.4. The scientists have also used. I'm not really going to punch my chihuahua. Scientists have also used human lymphocytes to make MABs to other pathogens and to some types of cancer cells. Suggests one reason why the new MABs have been more successful in treating diseases in humans than the MABs in mice. So the MABs that were made from humans. Right, so MABs made from human lymphocytes, MABs made from human lymphocytes are less likely to be rejected as they have similar antigens. Okay, so human antigens are going to be more similar on the lymphocytes on the MABs than on the mice. Mice are different. So mice are different animals, so they have different shaped antigens. They're more likely to be rejected. Okay, cool. All right. That's the end, folks. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. The exams are coming. Work your way through as many walkthroughs as you can. Use all of the internet resources out there. Use Physics and Maths Tutor. The website's fantastic. Use Bite Size. They've got multiple choice questions on the GCSE Bite Size. That's decent as well for just your memory recall. Make sure you sit at least 3 full pass papers timed at home for each of the science exams. So for your B1 exam, make sure you're going in there knowing that you have sat 3 past papers. So minimum 3 full past papers, timed them. You can work out your percentage. Generally, if you're getting over 70%, you're going to be smashing that level 9. All right, guys, I'll catch you on the next video. Take care.
You're still here? it's over.